evening to everyone. Today, as part of the continuous education, Unit 3, the animal nutrition will be studied. So, in that particular chapter, we are going to study about the nutritional requirement of the dairy cattle, especially the ruminants. So this is also one of the important thing that one need to know, especially the particularly what we need to study is what we need to study is the ruminant digestion, how it happens, the protein metabolism, how it takes place, and also the different types of the digestive system, especially which digests the proteins, especially in case of the ruminants are concerned. So coming to the first one, that's the especially the protein digestion in general, it involves the especially the denaturing the peptide bonds, and at the same time the it is going to release free amino acids. Free amino acids are released. The peptide bonds are broken and free amino acids are being released. So, because of this thing, then what happens is the protein digesting enzymes are either endopeptidases or exopeptidases. There are two types of these things are there, endo or exopeptidases. So the endopeptidases break peptide bonds within the primary structure into smaller fragments, whereas the exopeptides cleave amino acids of the terminal end of the protein molecule. Whereas the carbohydrate peptides, carboxypeptidases, removes the amino acid from the end with a free carboxyl group, and aminopeptidases act on terminal amino acid with a free amino group. So wherever the amino group is there, either the endopeptidases or exopeptidases or carboxypeptidases. The three things are being utilized. So here it is the especially the here we can see that different enzymes, protein digesting enzymes are the number one, the protein digesting number one, especially the endopeptidases. Then another one is the exopeptidases. The third one is the carboxypeptidases. Then the aminopeptidases. These are the enzymes one need to remember, especially the protein digestion is concerned. Okay. Then the coming to the next slide, this is the protein digestion mechanism, especially in case of ruminants. In a nutshell, what we can say is the dietary proteins are divided into rumen degradable proteins. One is the rumen degradable proteins, which are degraded in the rumen. Then another class is rumen undegradable proteins. So you know it very well. Then the classification of rumen degradable proteins are, some of the proteins are there which can rapidly degrade in the rumen, and some proteins are there which slowly degrade in the rumen. So then rapidly degrading proteins can be divided into ammonia, are converted to ammonia. Then ammonia is either directly going to the blood and going to increase the pH of the blood, especially, and sometimes if it is too much, then non-proteinous nitrogen may cause the toxicity. Whereas the ammonia and amino acids, the urea may be produced, then urea is going to the kidney, then it is going to be secreted in bladder through then to urine. Whereas the amino acids, which are produced both from slowly degrading proteins and rapidly degrading proteins, they are substrates for the microbial proteins. They become the microbial proteins. Whereas we will see what happens if a, especially what happens if a rumen degradable protein is being digested. So the intestinal proteins, then they become the rumen degradable proteins, they become the intestinal proteins, then they, some of them are digested or some of them are undigested. There are two types. So they are not bypassing the rumen directly, they become the substrate of rumen. Then some of them are digested, some of them are not digested. The undigested part of this, particularly the proteins, are excreted in the feces and they are not utilized by the ruminant. Hence, they are totally a waste. Then digested means they will go to the liver in the form of the amino acids. Then they will be utilized very well for the body tissues and also to form the milk proteins, which is very very nutritious in nature. Hence, the dietary proteins are two types. One is rumen degradable protein. So some of them are rapidly degraded, and some of them are slowly degraded, and both of them are converted to amino acid and ammonia. Then the ammonia which is generated here is going to directly absorb into the circulation and may increase the blood pH, and sometimes it is also converted to urea. Urea goes to the kidney and bladder, and also the urea will be metabolized by the liver to form certain proteins. So the rumen undegradable proteins, they are digested in the rumen itself due to the help of so many enzymes. Then they will go to the rumen, especially then they get digested in the rumen and absorbed into the circulation as amino acids. Then they form the good milk. This is the important thing to note as. So coming to the protein digestion is begins in the stomach itself. In case of most of the animals, the stomach itself or the monogastric animals. Stomach here means rumen, reticulum, omasum, and the abomasum. Then an enzyme known as the gastrin, a hormone, it's not an enzyme, it's a hormone, initiates the breakdown of proteins in the stomach itself, especially in the monogastric animals. So what happens? The presence of food in the stomach leads to the secretion of pepsinogen by the chief cells into the gastric mucosa, especially in case of the monogastric animals like the humans or any other animal. So the pepsinogen is activated to form pepsin. Then whenever there is a increased HCl in the stomach, which is produced by the gastric parietal cells of the gastric mucosa, then pepsinogen is activated. So in young animals, the milk coagulating renin is secreted into the stomach for the formation of the clot, which aids in transport into the small intestine. So this is the process wherein which the gastrin initiates the breakdown of the proteins into the stomach. Then in the stomach it leads to the secretion of pepsinogen. Pepsinogen is activated to form pepsin. Pepsin is going to digest the endopeptidase. Is an endopeptidase. So it is going to digest the different type of the amino acids. So this is the process how in case of a simple stomach animals the protein digestion takes place. The next portion of the digestion occurs in the small intestine, where in which the varieties of the enzyme are secreted. So the hormone named as the secretin is a hormone in the duodenum. It is secreted in most of the monogastric animals or the polygastric animal. It stimulates enzymatic secretions from the pancreas, which includes three active forms. Three active forms. So here the digestion occurs in the small intestine, by the action of the secretin in the duodenum. It stimulates enzymes, mainly trypsinogen, then chymotrypsinogen, and the another enzyme. Here I can highlight this thing, especially the trypsinogen, then chymotrypsinogen, then procarboxypeptidases. So these are the enzymes which procarboxypeptidases. So note down this enzyme: trypsinogen, chymotrypsinogen, and procarboxypeptidases. Whereas coming to the next one, that is the enterokinase is one also secreted. This is also another enzyme at the duodenum which converts trypsinogen into trypsin. So here it's also related, which then converts chymotrypsinogen and procarboxypeptidases to their active form, like chymotrypsin, which is mentioned here, and carboxypeptidases. So these are the important enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidases, enterokinases. Then the different enzymes are being secreted. So here another flow chart which you can easily understand how a protein metabolism in take place is: the dietary protein is supplied in the form of the crude protein, especially it is also the crude protein. Totally it is CP. You know what is a CP? It is also containing the water, and it is derived from the cakes of the grains. So the digestion takes place both in rumen and the small intestine. These are the two important places where the protein digestion takes place. Then the saliva is being mixed with the dietary crude protein. Then the it is converted to the true protein, especially it is engulfed and saliva is mixed. Then true protein is acted by the peptides. Then it is converted to amino acids. Then it becomes the substrate for the microbial protein. Then microbes are directed. Then usually rumen proteins are being produced. So these RUPs are metabolizable proteins, absorbed amino acids. So they become the endogenous proteins, then they become the building block of the body for the production of the muscles and also the milk fat. Whereas there is another role of the urea in case of the ruminants. Whenever urea is administered to the extent of 2% of total diet, then what happens is it is a source of non-proteinous nitrogen, whereas the cakes etc. are true proteins. These are not non-proteinous nitrogen. Then the non-proteinous nitrogen are converted to ammonia in the stomach, and they are going to the liver. Especially this is a very special process in which what we can say, especially in case of the ruminants, this is a process. So it is going to the liver. In liver, there are varieties of the enzyme which convert the ammonia to amino acids. So this is a process. Or urea is then secreted as amino acids in the mammary gland. So non-proteinous nitrogen are very cheap sources of the proteins, and which are very much required for the digestion of different, especially the production of cheap quality protein. But they are to be put only in small quantity, or else they are end up in the toxicities. So these are all the protein metabolism in case of the dairy cattle. Then there are varieties of the enzymes, the site of production and active forms. So the example is, to remember, the pepsin is produced in stomach, the place where pepsin is produced under stomach under HCl, this thing, especially HCl. Whereas the enterokinases are produced from the duodenum. Trypsinogen is produced in pancreas in the inactive form. Then it is converted to pepsin, trypsin in the small intestine. Trypsinogen is converted to trypsin. Whereas chymotrypsinogen, it is also secreted from the pancreas. It is also inactive form. Then it is also converted to chymotrypsin by the trypsin. So this is the trypsin. This is going to act on the chymotrypsinogen and it's going to be converted to trypsin. Then another enzyme that's called as the procarboxypeptidase. It is also secreted in the pancreas. It is also inactive, whereas it is going to be converted to carboxypeptidases. Procarboxypeptidase is converted to carboxypeptidases. Then by the chymotrypsin which is produced here, it is going to act here then in the small intestine by the trypsin. So these are all the procedures especially which are going to take place in case of the digestion of the amino acids, especially different proteins are being digested. So protein digestion in ruminant animal is mainly divided into two phases. One is digestion in the reticulorumen, reticulorumen, because this is one of the biggest organ. And the second phase is in the abomasal and small intestine. So two phases, it is divided into two phases. One is digestion in the ruminoreticulum where the microbial digestion takes place. Whereas the digestion in abomasum and the small intestine, that's the two parts. Therefore, the ruminant animals dietary proteins are classified as rumen degradable protein because it is going to be degraded in the rumen. Here it is degraded into rumen, hence they are known as rumen degradable proteins. Whereas the rumen undegradable proteins are also there, then they will go to the abomasum and small intestine. So simply this is the procedure how it takes place, especially in case of ruminants. So very similar to that of the monogastric animals, the main goal of the protein supplementation is to provide amino acids. Ultimately, we need to have the amino acids which are the building blocks of the proteins. Amino acids need to be produced. Whatever form of the protein is there, it may be a rumen degradable, non-degradable, crude protein, or whatever it may be. Ultimately, it need to be converted to the amino acids. Totally, it need to be converted to amino acids. Hence, the ruminants proteins serve as the source of nitrogen in the form of the nitrogen. Directly, they are not going to convert it to the amino acids, but they are provided as nitrogen to the rumen bacteria so they can make their own microbial protein from the scratch, especially the which is the material. So microbes do not care where the nitrogen source comes from. So this, remember please this one, the microbes will never care from what is the source of the protein where it's coming from and can use non-proteinous nitrogen substances such as urea for microbial protein synthesis. So this is the biggest advantage in case of the ruminants. They can convert non-proteinous nitrogen into the amino acids. So the urea is 100% degradable in the rumen by microbial urease. So this is urea is converted to the ammonia, but it becomes highly toxic when it is in the higher quantity. Hence, one should be very careful. It should not exceed the maximum quantity of 2%. Always it should never exceed or else it becomes toxic. So because of this characteristic feature, the proteins especially they are divided into different categories. Then they are always subjected to the degradation by the rumen by microbial urease especially, then that can be toxic at higher level. And the rumens, the protein entering the rumen by the way degraded by both bacteria and protozoa. So what are all the things? Even though bacteria are also participating in the carbohydrate digesting system, so which produce proteolytic enzymes. So these are going to produce the proteolytic enzymes. So protein entering the rumen, any type of the protein, is degraded by both bacteria and protozoa which produce proteolytic enzymes. So the bacteria are going to produce the proteolytic enzyme and they are going to be degraded. So the rumen microbes provide protease enzyme and peptidases to cleave peptides bonds in the polypeptides to release the free amino acids from the protein. So rumen microbes, they also produce the proteases and cleavage of the peptide bonds in the polypeptides to release the free amino acids from the proteins. And apart from this one, several factors such as the solubility and the physical structure of the protein can affect the rumen degradation. So many type of the proteins, for example it's a soya cake, then suppose there is the cakes like the groundnut cake, or sometimes the cakes are also from cottonseed cake. So these are also the important source of the proteins, but what happens is the solubility of all these is different in different type of the proteins, especially the proteins. So rumen degradable, rumen bypass proteins are always seen in different animals are concerned. Hence we need to see which type of the protein it is, then how it is going to be degraded. Then later another thing is these rumen degraded amino acids release ammonia, and the C skeleton of this protein process is called as deamination. So this is another important process where in which the rumen degraded amino acids release ammonia. Most of them are ammonia. Then along with the VFAs, volatile fatty acid, acetic acid, butyric acid and propionic acid. So rumen microbes synthesize their own microbial protein. This is a very important step which serves as a primary source of protein to the host ruminant animal. So the bacteria are going to produce the amino acids which is required for them, but the same is also serves as the primary source of the protein to the host ruminants. So the microbial protein is enough for the maintenance of the survival, especially suppose if even though the animals are not given with the very good protein sources, but they can even survive, but not high producing animals. So the deficiency of the protein takes place, and especially in case of high producing animals, we need to supplement with that of the cakes. That is very must. Whereas the ammonia absorbed from the rumen is converted to urea, and the urea is secreted into the blood as blood urea nitrogen or BUN. Ammonia absorbed into the rumen, then it is converted to urea, then secreted into the blood, and blood urea nitrogen is formed. Then coming to the next step is urea can be filtered, especially urea can be recycled, and urea can be recycled and filtered and recycled into the rumen via saliva or through the rumen valve. So whenever you are going to supplement the urea, it can be filtered and recycled into the rumen via saliva or through the rumen valve. So whenever the urea is administered, it is circulated, absorbed into the blood and secreted into the rumen to make the rumen pH alkaline, because the saliva pH is always alkaline. So the concentration of BUN in the ruminants reflects the efficiency of the protein utilization, how much quantity of the protein is being utilized. But one thing one need to remember that not all proteins are degraded in the rumen. This we need to remember. Not all proteins are degraded into the rumen. So that's one need to remember. And proteins that are not degraded by the rumen microbes are called as the bypassed proteins. So proteins that are not degraded by rumen microbes are called as bypassed, or they can also be caused as the what you call as the bypass proteins. What is a bypass protein means? They are bypassing the rumen and directly entering the abomasum. So proteins that are not degraded by the rumen microbes are called as the byproteins or undegradable proteins. Rumen undegradable proteins, or RUP, rumen undegradable proteins, and have a low rumen degradation rates, and have a very low degradation rates. For example, the protein in the corn is not digested in the rumen. It enters the abomasum, then it enters the intestine, then so many enzymes like chymotrypsin, trypsin, etc. are going to act on them, then the amino acids are released. So hope you know the concept of bypass proteins, because the bypass proteins are the one which bypass, means they cannot be digested in the rumen, and directly they will go to the abomasum and get digested there. Rumen undegradable proteins are, and they are entering the abomasum, and later they are going to be small intestine of the ruminant and animal for the digestion and absorption. So proteins reaching the small intestine could become rumen undegradable or rumen unutilized proteins, or those from the those from the microbial source. Both of these can be utilized especially. So the point to be taken here is not all proteins are degraded in the rumen, and some proteins are not degraded in the rumen, are going to the abomasum, then they are going to the intestine, then later in the downer rates they are going to be absorbed into the circulation through the different enzymes which are converting them into the amino acids, and later they are going to be absorbed into the circulation as amino acids. Then the amino acid needs the host animal are met by the rumen undegradable proteins. So the amino acids, total amino acid requirement of a cow which is producing about 10 liters of the milk, is always supplemented by the microbial digestion proteins which are digested by the microbes. So there is not a problem here. Whereas for the additional milk yield, we need to supply the rumen bypass proteins. So both ruminants and monogastric animal, they require the essential amino acids in their diet. You know what are all the essential amino acids like methionine, cysteine, etc. Then the amino acids cannot be stored within the body. Another thing is the amino acids, whatever it may be the form, they cannot be stored in the body, stored within the body. Amino acids cannot be stored within the body. So a continuous supply of the uh, diet, protein diet through the diet is very much required. Hence, always the proteins are continuously, they need to be supplied, because they cannot be stored in any other form like the fat, etc. So they need to be supplied by the dietary supplementation of the proteins. So the some of the similarities and differences in the monogastric and the ruminant animals are there, because there is a pattern in ruminant, and there are some differences how the protein degradation takes place in the rumen and non-rumen especially. That is the difference between the monogastric animals and the ruminants, differences especially. So what happens is, in case of the amino acids in the monogastric animal, their amino acid profile at small intestine reflects the diet. In case of monogastric animal, the amino acids which are absorbed from the small intestine is the only amino acids which are available to the body. Whereas in case of the ruminants, amino acid profile at small intestine is different from that of the diet, because many times the rumen also takes place, especially the ruminant digestion also takes place here. So here there is a difference between the amino acid, and no upgrading of low quality protein, especially in the amino, especially the monogastric animal. The amino acids which are pointed, especially, are always like that. There is no change in them. Whereas the upgradation of low quality dietary protein is always present in the ruminants, and that is the capacity of the ruminants to convert the non-proteinous nitrogen like urea into the amino acids. Whereas the monogastric animal, the protein quality is not downgraded. The protein quality, especially the different amino acids, need to be produced, whereas the downgrade high quality dietary protein. So these animals do not require the high quality dietary protein. Whereas the monogastric animals cannot use non-proteinous nitrogen like urea. They suffer from toxicity. Whereas the ruminants, they are able to use NPN, especially the urea, for the production of the ammonia and subsequently the amino acid. Then there is constant supply of amino acids are always required, especially in case of the monogastric animals, for the supplementation of the proteins. For example, a human being has to eat the egg always, or the meat, or the milk, whereas constant supply of amino acids is required in case of the ruminants also, but not in the form of the pure protein, because here even with the raw crude protein also, these animals can produce their own proteins or amino acids. So this is the difference, basic difference between the monogastric digestion and the ruminant digestion of the protein in nature. Then here there is a diagrammatic representation of the degradation of protein. Here it is unprotected protein and unutilized protein. Then protected protein means the structure is little bit different. Then what happens is, the protected proteins, they enter the, especially the duodenum, and they undergo the digestion in the intestine. Then in the form of the amino acids, they are going to be absorbed into the circulation. Whereas the unprotected proteins, or rumen degradable proteins, they are converted to ammonia and the amino acids, and this will be utilized by the microflora. Then some of them get lost, and ultimately in the rumen itself they are going to be absorbed. Whereas the protected amino acids, or the bypassed amino acids, or the proteins, they directly go to the, especially the abomasum, then intestine, then they are going to be digested. So this is the basic difference. Then what's the concept of the bypass potential of the protein supplement? And many times we are hearing that the bypass proteins are very much required to the high yielders. What is the reason for this thing? Always we need to supply the bypass proteins. Once again, the proteins are classified into the rumen degradable proteins and rumen non-degradable proteins. So these are also called as the rumen bypass proteins. So among the cereal grains, there are the proteins are the source from the cereals, grains. Corn has the highest bypass potential. the corns, maize corn and other corns. So it has got the highest bypass protein. However, it should be noted that corn is deficient in essential amino acids such as the lysine and methionine. So you have to know that the corns are deficient in essential amino acid like the lysine and methionine. So corn is a bypass protein, but whenever we supply this corn, then there is a chance of the lysine and methionine. Hence, we need to supply the other source of the protein. So, whereas the animal protein sources such as fish meal, of course, not used in case of cattle, but only used in case of the poultry, and meat meal have high bypass proteins. Meat meal, which of course these are used in case of the poultry and sometimes the pig. So these are highly using bypass potential. And drying forages and heat treatment increases the bypass potential. Means if you dry the particularly the cake making, and if it is dried properly, then or if you are sterilizing it with the heat and subjected it to the high temperature, then the bypassing will be increased. That's the thing. And the here also there is one chart how the proteins are going to enter the rumen, and certain terminologies you should understand that RUP means rumen undegradable proteins. RPM means rumen protected methionine. RDP is rumen degradable protein. NPN, as usual know, non-proteinous nitrogen. MP is microbial protein, ammonia, amino acids, then mammary gland, then undigested protein. So these are all terminologies, so that we can understand this chart very properly. So rumen undegradable proteins, or they can be called as rumen protected methionine, which are containing rumen uh, protected methionine. They also enter. Then rumen degradable proteins are also entering. Then non-proteinous nitrogens like urea, etc. They are going to enter the ruminant system by the different feed sources, especially. So what happens is the feed source will be like that. So the non-proteinous nitrogen in the form of the urea is recycled in the saliva, recycled in the saliva. So whenever the urea is administered, so it is immediately absorbed into the circulation, then it enters the circulation, secreted in the saliva, and makes the alkaline pH of the rumen. So now we are here, then slowly let us enter inside the rumen, then the proteins, where the rumen degradable proteins, they will go to the place where they get degraded by the ruminal microflora. Then the amino acids like ammonia are formed. Whereas the non-proteinous nitrogens, they are also becoming the substrate of ruminal microflora. Then they become the source of the, especially the ammonia. They become the source of the ammonia. Then the excess urea that is absorbed into the circulation, that is either secreted in the saliva or go to the kidney and excreted out of the body. So the blood urea nitrogen concentration in ruminants is very high when compared to the other animals, and that is why the ruminants are the one which are always utilizing the, especially the rumen degradable proteins. So the rumen undegradable proteins. So here you can see that they are settling down and they are converted to metabolic microbial proteins. Sometimes They are also becoming the substrates of the microbial protein. Then they are entering the intestine and abomasum. Intestine and abomasum. And here lot of the enzymes are present and they are going to convert them into the different amino acids. So these amino acids are entering the liver and getting converted into the different types of the proteins, especially they are called as the milk proteins, and they enter the mammary gland and secreted through the milk. And the undegradable proteins, once again, they are undigested proteins. Totally they are going to be secreted in the feces. That's how the particular structure one need to know. So once again, all these are rumen undegradable proteins, rumen protected methionine, and rumen degradable proteins, and non-proteinous nitrogen. All are entering the rumen, whereas the rumen degradable proteins become the substrates for the ruminant microflora. Then they become the rumen degradable amino acids. The ruminant microflora are going to produce the ammonia. Then ammonia is also converted to urea. Urea is recycled via saliva and urine. Then the non-proteinous nitrogens become the substrates for the ruminal microorganisms, and they are going to produce the ammonia. Ammonia is utilized as the one of the source of the protein, or it is also called as the microbial protein. Then ruminal protected methionine or the ruminal undegradable protein always enter the intestine and abomasum. They become digested and getting converted to amino acids. The amino acids are entering the liver and converted to milk protein and secreted through the milk. And anything which is secreted or which is digested is also utilized for the formation of different quality of the proteins which is required by the cattle. Then what is the bypass concept? You know that what is a bypass concept is this one. Here the proteins are not being substrates of the ruminal microflora. They enter the abomasum and intestine, getting there degraded into amino acids, and fastly they are going to form the proteins. Hence, this is the concept of bypass potential of the protein supplements. So there are varieties of the feed processing methods, such as one is the pelleting, then steam rolling or flaking tend to denature the feed protein due to the generation of the heat. Yes, this is also another thing, but they thereby protecting the protein by lysis in the rumen. So sometimes the proteins are lysed in the rumen, and such type, especially the pelleting, many times the feeds, the cattle feed is made as a pellet. Then pellet is subjected to the steam rolling or flakes are made, and they tend denature the feed protein, and due to the generation of the heat, thereby protect them from the protein lysis in the rumen. Then number one, whereas the rumen protected protein sources through formaldehyde treatment is also another method that remain intact in the rumen and dissolve in the abomasum are commercially available. So this is also this thing. For example, some fish technologies are there which make the rumen protected protein sources through some formaldehyde treatment that remain intact in the rumen and dissolve in the abomasum, and are commercially these are available as bypass proteins. But the thing is, the formaldehyde concentration should be in the limited. That's the concept of the bypass proteins. Bypass proteins are very essential, especially in case of the animals which are producing very high quantity of the milk for immediate supplementation of the proteins in the form of the amino acids. Hence, there are few of the key points are there to take home. Then the digestion of the protein starts in the stomach with hydrochloric acid, wherein which the acid denaturates the protein. Then the another one of the enzyme is there, that is called as the pepsinogen. Pepsinogen, it is converted to pepsin, active formed by the hydrochloride HCl. So we know this thing. So this is the digestion of the protein in the monogastric animal. Pepsin cleaves proteins to form the peptides. Pepsin cleaves proteins to form the peptides. Peptides are further cleaved by the different enzymes in the intestine. Then they are going to form the amino acids. So the small intestine has got several enzymes as we have already listed. The pancreas releases trypsinogen, chymotrypsinogen, and carboxypeptidases. So these are the enzymes which are present in the small intestine of both ruminant animal and also the non-ruminant animal. Then apart from this one, the enterokinases, these are secreted from the, especially the enterokinases are secreted from duodenum and convert trypsinogen to trypsin, which then converts chymotrypsinogen to chymotrypsin. So it's a cascade of events. Then procarboxypeptidases to carboxypeptidases. So this is a cascade of events especially. Then degradation by pancreatic and small intestinal enzymes results in amino acids are formed, which are the building blocks of the peptides, and the di and tripeptides are always formed. So this is the way how the proteins are formed in the body. Then absorption of the villi, which is in the intestine, there are villi are there. Then microvilli occurs using the carrier proteins. Carrier proteins are there. They are going to pick up certain amino acids and also the energy. Then absorption is affected by the nature of the amino acids and some whole proteins, and di and tripeptides are also absorbed into the circulation. Whereas it is a little bit different process in case of the ruminants. Then ruminant microbes release enzymes like proteases and peptidases, ruminal microflora. They are going to release the different enzymes like proteases that cleave peptide bonds and release amino acid directly. So this is the thing, capacity of this, so that they are going to have the proteases and peptidases. The ruminant microflora, they secrete certain enzymes like proteases and peptidases. So they cleave peptide bonds and release amino acids. So the microbes then deaminate or remove amino group and amino acid, releasing ammonia. So they are going to act on the peptides and deaminate and remove amino group from the amino acids, releasing ammonia and the carbon skeleton. So microbial use the ammonia. This is the capacity of the microbes to utilize the non-proteinous nitrogen like urea. Then the carbon skeleton and energy in the form of the volatile fatty acids to synthesize their own proteins, their own amino acids or the protein. So this is the beauty of the ruminant digestion system. So ruminants has no amino acid requirement actually. They can convert the non-proteinous nitrogen into the amino acids. So they have a nitrogen requirement. They require simply the nitrogen. They do not require any costly proteins. But the ruminants break down dietary protein into the ammonia and carbon skeleton through rumen microbes and synthesize their own microbial protein. So because of this thing, they will fulfill the non-proteinous nitrogen also, maybe in the form of the urea, or even though the protein quality is very low, the ruminant microflora are going to produce their own amino acids. But therefore, a portion of the ruminant's protein requirement can be met with non-proteinous nitrogen. That's why this capacity of the ruminant microflora to convert the non-proteinous nitrogen to the ammonia that is utilized as one of the chief source of the protein. So urea is the best example of the non-proteinous nitrogen. Urea is going to become the very good source of the non-proteinous nitrogen and a readily available carbohydrate source. It's readily available carbohydrate source to provide the carbon skeleton. See, we require two things. One is non-proteinous nitrogen and a carbon skeleton in the form of the fiber, maybe a crude fiber to protein synthesis is a critical event, or else the toxic ammonia builds up quickly in the rumen. Hence, whenever we are supplementing the urea, very good quality fiber or the hay need to be put, which provides a carbon skeleton to the microbial digestion, so that the proteins leaving the rumen are microbial proteins, and those that escape the rumen degradation are called as the bypass proteins, and these are the proteins that are not extensively degraded in the rumen. So the ruminants have got a very characteristic feature of the two type of the digestive system. One is and the rumen and reticulum, and the remaining is the abomasum intestine, maybe the small and large intestine. So the feed processing can affect the bypass ability of the protein. For example, if the cakes are made by heat process or the formaldehyde application, then they can be converted to bypass proteins, especially so that they can be utilized for the ruminants for production of the quality amino acids. Then so this is about the very simply the digestion process of the especially ruminant digestion system. So they have got the protein nutrition not available to the non ruminants. Non ruminants cannot digest the protein so easily, and non ruminants always require the very high quality protein, whereas the ruminants can digest the low quality protein and produce high quality amino acids in the form of the rumen microorganism. So the required energy is derived from the plant polysaccharide, as you know very well, because the uh, proteins, especially the plant proteins, polysaccharides, are being degraded by the ruminant microflora as cellulose and nitrogen is derived from the ammonia and amino acids in the rumen. So all these things, the cellulose is providing the carbon skeleton, and ammonia and amino acids in the rumen. So this microbial protein is directly available for digestion and absorption by the host animal, and the principal source of the amino acids to the ruminants. So they do not require the extra protein in toto, but whenever there is a high protein, high milk yielders animals, then we need to give them the exogenous proteins. So, cow can maintain unimpaired milk production on diets lacking protein also. Even the proteins are not there, means no good quality protein is available. Even then, the ruminants can survive because they have got the nitrogen metabolism, which is usually regarded as being insufficient. So dietary protein is broken down much rapidly relative to the breakdown of the energy containing plant fiber results in excess ammonia production. So this is also required. Then bulk of ruminant feed is composed of vast variety of the nitrogenous compounds along with the nucleic acid association with protein from plant source, and also the ammonia etc. may be present. And nitrogenous input in the form of the urea etc. to the rumen fermentation or salivary mucoprotein in urea, then later enters both saliva and by diffusion through the rumen wall. Then this is once again the rumen degradation protein where they are substrates of the ruminant microflora. Then the ammonia is produced, then it's going to form the amino acids. So here you can see that the protein nitrogen utilization by the ruminants. So this is the crude protein in the diet or rough protein. So there are once again two proteins and non-proteinous nitrogen. Both are entering the rumen, and the 60% is converted to the amino acids here. Then they are converted to keto acids, whereas the non-proteinous nitrogens in the form of the urea, they enter the urea in the saliva etc. Then they are converted to ammonia. Then the ammonia is always utilized by the bacteria to produce the microbial proteins, whereas the carbohydrates, they provide the carbon skeleton in the form of the volatile fatty acids, and the both fat and the proteins, they form the part of the energy, whereas the diet protein that escapes the microbial digestion is called as the bypass protein or rumen undegradable protein and escape the protein, and this will be digested in the intestine, and the one which is not digested by the intestinal enzymes will escape and they will be secreted in the feces. So the urea is excreted ultimately whenever it is not utilized by the animal, it is excreted into the urine. Then the phase of the digestion of the protein is rumen phase, and another one is the intestinal phase. So all these interconversions are generally considered to occur by the action of the different microbial enzymes like the peptidases etc. So microbial protein is the most abundant form of the protein of living the rumen, especially microbial protein. So endogenous urea will only be quantitative significant for microbial protein synthesis. So endogenous urea will only be the quantitative significance, especially for the microbial protein synthesis when dietary protein is very very low. So even with the dietary protein is very low, they ruminants can digest or produce their own protein because the non-proteinous nitrogen compound, non-proteinous nitrogen compounds are also, dietary protein is low. Ammonia is the major product of the catabolism and also main substrate for microbial protein synthesis. So, the ammonia is overflow leads to the inefficient nitrogen retention. Then breakdown of the dietary protein is degraded by the microbial enzymes which are present in the rumen, like the secondary or tertiary structure, and cleavage of disulfide bond enhances the breakdown, similarly heavily cross-linked proteins. So introduction of artificial cross-linking into the protein inhibit their hydrolysis, like the heating of and carbohydrate, this formaldehyde treatment, this is the one of the method to make the bypass proteins. So the both of these process will reduce the solubility and cross-linking, thereby producing the bypass protein to the lower tract. However, these treatments may impair the subsequent availability of some of the amino acids which are lysine, cysteine and tyrosine because they are heat labile and degraded. Then nature of the diet has a major influence on the proteolytic enzyme of the rumen content, and fresh herbage promotes the activity up to 9 times than that of the dry rations. And cereal diet is yielding the higher proteolytic activity than that of the dry forage because the proteolytic rumen microorganisms tend to be amylolytic rather than the cellulolytic. So the rumen proteolysis probably varies from protein to protein with other constituents of the diet. So here one of the model is also depicted here with the endogenous protein is produced by the feed protein and also feed non-proteinous nitrogen. So the feed protein is also converted as endogenous feed. Then non-proteinous nitrogen is converted to ammonia, especially in case of the rumen. Then the ammonia is circulated into the blood and enters the liver and gets the converted to the urea, and some part is also utilized by the ruminant microflora to produce their own protein, whereas the feed protein, degradable protein, so this is also called as the microbial protein. So it is going to pass into the protein, endogenous protein and bypass protein and microbial protein. Whereas the non-proteinous nitrogens, they are converted to once again into the ammonia. Then varieties of the enzymes, proteolytic enzymes are present in the intestine, then also the abomasum and everywhere to convert the protein into amino acids. So varieties of the ciliate protozoa are there which are going to break down the absence of exogenous source of nitrogen and digest the rumen bacteria. And many entodinium, including the different types of the protozoa, are also proteolytic in nature. Mixed protozoa and much higher aminopeptidase activity than rumen bacteria, slight higher trypsin activity. So, the main role of the protozoa is hydrolysis of the particulate proteins to appropriate particle. And predominantly, the activity of the protozoa against rumen bacteria is of enormous significance in the bacterial turnover, especially in the rumen. And if the protozoa are absent, then bacterial protein turnover varied from 3% to 2.7%. So that is going to be decreased. So main role of protozoa is not to hydrolyze the exogenous protein, but in metabolizing bacterial protein which needs hydrolytic activity. So apart from this one, different bacteria are also involved in the digestion of the protein. So the proteolytic organisms are Ruminobacter, Amylophilus and many more, and these do not require peptides or amino acids for the growth even available. Ammonia remains the principal nitrogen source. So that's non-proteinous nitrogen source is enough to be labeled as the protein and also peptidases. So ultimately what we can understand is, even though the ruminants can survive without the proper or good quality proteins like very good cakes etc., unlike the monogastric animals, especially the monogastric animals do require the original protein and they also require the quality protein very high, whereas the ruminants, as they do have lot of microflora, they have got the protein breakdown capacity when compared to the non-ruminant are concerned. Hence, the ruminants have got their own digestive system, which is has got made them as a characteristic animals for the formation of the amino acids and peptide amino chain, then especially dipeptidal, peptidases, etc. are there. So ultimately what we can understand from these different slides is the microbial population, if it is altered, then there is a response that there is deficiency of the proteins also. So the ruminant microflora need to be very well enriched to get the metabolism of amino acids in the next stage of the metabolism and most of the constituents of the digestive dietary protein. So, and ultimately the proteins need to be used by the ruminants, and ultimately they are going to produce the different types of the amino acids. They may be in the form of the amino acids and also the peptides and proteins. So this is about the digestion and utilization of the proteins by the ruminants, and thank you very much for patience hearing, and thanks a lot for being with me.